of Chapter 11. Chapter 11, we learned the physical, chemical methods of controlling these microbes. Chapter 12, here we're looking at the antibiotics. We're looking at these chemotherapeutic drugs, these antimicrobials. A little bit of history. So we were looking at 100 years ago. The stats were that 1 out of every 3 children were expected to die from some type of an infectious disease before they turned 5 years old. Okay, that is quite a scary statistic, 1 out of every 3 children. So here we're looking at 100 years ago, and then around the 1930s into the 1940s, this is when the first antibiotics were discovered. So Alexander Fleming, he was responsible for discovering penicillin, and it actually was an accident, the way he discovered penicillin. Again, that was Alexander Fleming. And what he was doing was he was growing some bacteria. I believe it was Staphylococcus aureus. I think he was growing Staph aureus on a plate, and he left the window open, and some mold spores gained access to the plate, and the mold spores happened to be from the penicillium mold, landed on the plate, and killed off a lot of his staph. So he's like, wait a second, something from the environment showed up and killed the bacteria. What if we can harvest the substance from the environment and produce it and then administer it to individuals to kill a bacterial infection? This is where it all began. So it was actually, kind of an accident where we discovered this miracle, this first drug, this penicillin. So 1930s into 1940s, all of a sudden this statistic, totally changed because now we have these antibiotics in order to treat these infectious diseases. Everything was awesome. And then all of a sudden things started to change. So here we're looking at, going into like the 1980s, approaching the 1990s. You didn't hear about it very much, but we're starting to see that a lot of these antibiotics were no longer effective on these bacteria. But we didn't hear about it because, okay, so antibiotic A isn't working on the bacteria. That's okay, because we have antibiotic B, and that worked on the bacteria. Awesome, we always had a backup. Nowadays, all right, A's not working, B's not working, okay, Q, R, and S aren't working, and then all of a sudden, we have nothing. Now we have nothing that works on some of these bacteria. So now we're at this, kind of approaching this era, which we would call the post-antibiotic era, where soon we may be having a number of infections. We just don't have any drugs to treat it. Okay? And this can definitely turn into, a world health crisis if you have some type of a pandemic and you can't, you don't have any medications to treat it. And this, bacteria, we talked about bacteria being here for what? 2.53 billion years, right? They've been here a really long time. They're not going anywhere. We can't expect them to be eliminated from our environment. They do so much good for us. Obviously, they're, adaptable. They can adapt to any situation. So the more and more that we toss these drugs at them, the more and more they're going to adapt to them and change and be able to overcome them. Okay, so this problem has gotten out of hand for one primary reason, and that is the overuse and also the misuse of antibiotics. Okay, again, the more you toss at them, the more they're going to change and adapt, and they can easily kind of spread this resistance to other bacteria. Okay, so it might start with one, but then every other bacteria in the environment in the near proximity could also be resistant as well. All right, so let's talk a little bit about these drugs. I'm going to give you some definitions. We'll talk about what makes a perfect drug. So ideal goal is this. Ideal goal is to administer a drug which is going to knock out the infection. So you want it to kill whatever organism is causing the infection, but you do not want it to harm the host. That is not easy to do. A number of infections where there's maybe 1 or 2 last-ditch antibiotics that still work against the bacteria. A lot of them are kept on reserve because they're so toxic. The patients are taking them because it's the only ones that work, but it's extremely toxic, where you're looking at it completely shutting down their kidneys or something else. So, it's really hard to do in a number of cases is to, have this drug which kills the bacteria but doesn't hurt the host. So what do you think makes a perfect drug? So if I were to ask Desiree, what would make a perfect drug? If you were to think about a perfect antibiotic, what would you say about it?
It would be 100% effective.
100% effective. Okay, so it's going to target what it needs to target, right? Okay, so it's going to target exactly, the particular organism it's supposed to target. And maybe it's going to leave everyone else alone. So we know that we have normal flora throughout our body. You don't want that drug to kill off your normal flora. You want it just to target that specific bacteria. Amanda, do you have another thing that would make a perfect drug?
Something that's available to everybody and affordable.
Yes, awesome. Affordable.
That was good.
Okay, so looking at something that's readily available, that's easy to get, okay, and it's affordable. I mean, that's not easy either. I mean, a lot of these drugs are very, very expensive. Okay, so you want something affordable. You also want something, if you're looking at a perfect drug, like what Desiree said, it's going to target that organism. I mean, you want it to kill that organism. You just don't want it to stop the organism from growing, but as soon as you stop taking the drug, then it can grow again. You don't want that. Does anyone remember from chapter 11 the name of that term? Microbistatic is when they're at a standstill. Okay, you don't want it microbistatic. You want it microbicidal. You want it to actually kill. So here's a few characteristics that would, be ideal. The ideal antibiotic, the ideal antimicrobial drug would be these characteristics. So selectively toxic. That means it's toxic to what's infecting you, but it doesn't hurt you. Awesome. What else do we have? Microbicidal. Yes, you want it to kill, you just don't want it to kind of keep something at bay. You want it soluble, you want it to work even though it's dissolved within your body fluids. Stays potent long enough. You don't want to excrete it too early. Okay, you want it to hang out and do its job. Here's a big one. Okay, it doesn't lead to the development of antimicrobial resistance. A huge topic we'll talk about today in the second half of the lecture. Works with your immune system. So it makes your immune system work better. It complements what your immune system is doing to target that infection. That's good. Stays active, gets to where it needs to go. Okay, so delivered to the site of an infection, reasonably priced, and doesn't lead to, there's not horrible side effects. Doesn't lead to an allergic response. Those are important things too. Okay, again, some of them can be quite toxic, and having a perfect drug, it wouldn't be toxic. So again, does this perfect drug exist that fits every single characteristic? We don't have the perfect drug, but we're trying. We have a number of really good antibiotics out there. But the organisms are really smart, and they can, find a way to combat these drugs. All right, so again, where do they come from? The antibiotics come from bacteria, and they come from fungi. Those are your two primary sources for antibiotics. Antibiotics, they have the term bio in the name. Okay, antibiotics. Okay, so it's got bio means life. Okay, so antibiotics are created from another living organism, from bacteria or from fungi. Examples I put up here. We have the Streptomyces bacteria, so that is used to harvest streptomycin. We have Bacillus. Okay, so bacitracin, you heard of bacitracin, you're looking at Bacillus. Bacitracin is from a Tracy strain of Bacillus. Okay, so bacitracin. Penicillium mold, of course, for penicillin and all the derivatives of penicillin. Cephalosporium, of course, used for the cephalosporins. And of course, a number of these drugs can be altered. They can just kind of change a chemical group and create new ones like amoxicillin or ampicillin, methicillin, and so on. Creating new drugs is tough, and it's really tough due to this antimicrobial resistance. It's a gigantic problem worldwide right now, coming up with new antibiotics. If we think about this for a moment and we think about antimicrobial resistance, okay, we think about all the bacteria that are resistant to these drugs. And you think about profit margins and all this. If you were a drug company, okay, and you had to produce a drug, maybe, if you produce a drug for hypertension, let's say, okay, or cholesterol, that particular drug can be used for years and years and years and years and years. Okay, it's going to be viable, useful, and so on. It's not going to change. You're going to make a lot of profit from that. If you were in a drug company and you were making an antibiotic, it takes probably about a decade, okay, to produce, kind of come up with this formula, produce the antibiotic, go through trials and stuff like that. It takes a really long time, costs a whole lot of money. That drug finally goes on the market within a year. About half of those organisms built up resistance to it. A few years later, just about every organism has built up resistance to it. So you spent more time and money in the lab making the drug than the drug was actually out there. So how much profit is there for drug companies making antibiotics? Not as much as, compared to the ones if you were in the market for making, something to reduce cholesterol. Okay, so getting drug companies to produce antibiotics is tough. So World Health Organization, there's a few organizations trying to just kind of give incentives to drug companies to continue to do this. It's very, very important. Okay, so let's run through some definitions. Let me kind of zoom in on this to make it less scary. Okay, number of definitions I want you guys to know, so table 12.2 in your textbook. First off, we have chemotherapy, just the raw definition of chemotherapy, using chemicals to kill. Okay, using chemicals as treatment, more specifically. Okay, so using chemicals as treatment, not to kill it. So chemotherapeutic drug is the drug which is used as therapy, is used as chemical treatment. Okay, so any chemical used in the treatment, that is the drug itself. Prophylaxis, that's a preventative measure. Okay, so prophylactic drug would be something you would administer prior to encountering or maybe going to an area where something is endemic. Antimicrobial chemotherapy. So again, chemotherapy, using a chemical as treatment. Antimicrobial means you're using a chemical as treatment against microbes. Okay, so chemotherapy is very broad. You're just using chemicals as treatment. Great. Antimicrobial chemotherapy means you're using chemicals as treatment, but against microorganisms, against infection and disease. The word antimicrobials, this is a big umbrella term. This is any type of a drug. Doesn't matter how it was made, how it was produced, where it came from. Anything which is going to work against microbes. Okay, anything working against microbes is going to fall under antimicrobials. Okay, the next three that I'll show you. Antibiotics, semi-synthetic drugs, and synthetic drugs. All three of these are drugs that are used to treat microbes, okay, used to destroy microbes, used as treatment when you have an infection. The difference between these three is how they're made. Antibiotics, again, it has the term bio in it. What does that mean? Life. Okay, so bio means life. So antibiotics, these are derived from some organism, whether it's bacterial, whether it's a fungi. If I skip one and go down to synthetic drugs are not created from microorganisms. They're created in a lab from chemicals and so on. Okay, so here you're looking at chemical reactions in a laboratory setting. Semi-synthetic drugs are a combination of the two. You take an antibiotic and then you chemically alter it. That's semi-synthetic. So these three are just, different by how they are made. That is it. Last two that we have, narrow versus broad spectrum. Narrow spectrum, you can think about it, I like to use the analogy of a sharpshooter, okay, because that narrow spectrum is going to pinpoint that exact microbe and destroy it and leave everybody else alone. Which, if you have an infection, you would want, and you knew, or your physician knew exactly what microbe was causing your infection, you would want a narrow spectrum antibiotic. Because you want to kill just that infectious agent, you don't want to kill off any of your good bacteria. Okay, so narrow spectrum is going to be the way to go, as long as you know what the agent is. Broad spectrum, you can think about this, not like a sharpshooter, but more like a bomb almost going off, in the sense that it is going to destroy what's causing your infection, but then it's going to destroy everything else as well. Okay, so it's going to destroy a very wide range of bacteria, which if you don't know what's causing the infection, or if it's caused by multiple bacteria, broad spectrum may be the way to go. If you know what's causing the infection, you want to get as narrow spectrum as possible. Broad spectrum, they can be quite dangerous. If you have, if you go into the dentist and you have a tooth infection, okay, they are going to give you a broad spectrum antibiotic. There's so many bacteria in your mouth, it's crazy. The bacteria that are infecting, okay, your tooth, it's going to be, this infection is caused by a handful of bacteria. They don't know exactly which one, there's a handful. So you're going to get something like amoxicillin, right? Amoxicillin is a broad spectrum antibiotic. Not only that, but you're going to get a super high dosage of it, like 1,000 milligrams or something. That's going to wipe out all kinds of bacteria in your system. Yeah, it's going to definitely kill what's causing the infection, but it might also kill good bacteria elsewhere in your body, and it might lead to a super infection. So you take something like this, and if it kills all the good bacteria in the vagina, for example, and then whatever yeast numbers are there can start to grow and then end up with a yeast infection. Okay, because the broad spectrum killed all the good guys. Or it knocks out a number of good guys within the gut, and then any pathogens that are there can take over, and it's going to cause, some pain, some diarrhea, and so on. Okay, so you can have a secondary infection take place in one of the intestines. So it wipes out a lot of the good guys, but that can lead to secondary infections. Yes,
Are the broad spectrum more harmful to children than adults as far as the risk of other infections occurring?
It's harmful to both.
Okay.
I mean, any of these would be more harmful to children in the sense that their immunity is still kind of developing. They don't have a very established normal flora yet. So wiping out that normal flora, and then having it reestablish itself, is still in the growing process, in that process. So yeah, a lot of these are. And the same thing, looking at the elderly where their immune system is compromised too, you're seeing a lot of the same scenarios. So some basic definitions. We'll talk more about this as we go through today. Okay, so starting treatment. So if you're starting treatment with a particular antibiotic, it's really good to know what's infecting you, right? Okay, so nature of the microorganism causing the infection. You would hope that your physician would run some tests and try and figure out what it is. Unfortunately, that doesn't always happen. Sometimes you're like, oh, well, I've seen, a handful of cases like that this week, so I'm going to treat it the same way. Hopefully it does the job. If it doesn't work, call me next week. That happens a lot. But knowing exactly what's causing it would be ideal. Degree of the microorganism susceptibility to drugs. That means how well does that particular antibiotic kill that bacteria? Well, think of the lab that we did on Tuesday, where you put bacteria on a plate and then you just stamped it with these discs. It's exactly what they do in clinics and labs throughout the country. Same thing. If your infectious agent that is infecting you, and they run a test with a handful of antibiotics and they see that those antibiotics do nothing, well, they're not going to prescribe those antibiotics. Okay, they're going to prescribe one which would result in a very large zone, which does a lot of killing. Okay, so you need to know that. And then, of course, looking at the patient. Again, children and the elderly, certain medications you want to avoid. Also looking at allergies, looking at kidney function, if they have any other, infections or diseases, that all plays a role too. Yes.
How do, let's say a kid, for example, develop allergies to so many antibiotics? Like my son can only have augmentin or vancomycin. That's it.
Those are the only two. And what's funny is we're seeing a lot more and more of that today than we did a long time ago. A lot of people will say it's from preservatives in food or from lighting, just from environmental factors. Allergies are one of those gigantic mysteries, where anyone can develop allergies at any time to anything. The rhyme or reason why, it's something that's unknown. But it's definitely something, it's something that some people can overcome in their lifetime. It's just how their immune system decides to react to something. But there's no exact reason why that happened to your son and maybe didn't happen to someone else's son. Some of it they say is genetics, but
Good job, Laura. Right.
but it's
Looking out.
allergies are definitely one of those big mysteries, And with allergies, first exposure, that's just when they're sensitized to it. Second exposure, that's where you see, the reactions. That's when you know they would be allergic.
Also discussed on the first time.
Okay, so identifying the agent, again, it's best if the physician runs a series of tests, right? That's your ideal scenario. It doesn't always happen. Oftentimes, like I mentioned, you're looking at the informed best guess of a physician, like, it seems like it's this, let's treat it as this, hopefully I'm right, An example I put down here would be an example where signs and symptoms may be enough. Further testing, is always recommended. But something like this, let's say a patient comes into the clinic and, has some pus from the genitalia. Okay, so something is definitely going on. They have, signs and symptoms of this gonorrhea infection. And so they take a sample and they just run a gram stain. And running a gram stain, along with the signs and symptoms, okay, with this pus, they see gram-negative diplococci. Gram-negative diplococci, like that combination, okay, a lot of gram-negative bacteria. There's a lot of cocci out there, okay, that are gram-negative, but there's so many more rods that are gram-negative. It's just kind of how it is. But diplococci, not very common that are gram-negative. Plus, you're not going to see gram-negative diplococci from the genitalia. So right off the bat, that along with the symptoms of gonorrhea, they can safely say that this would be a gonorrhea infection. Of course, further testing would be recommended, just to, say for sure. And with gonorrhea, there's like 3 or 4 infections where there are some of these multi-drug resistant strains. Gonorrhea is one of them. There's a really scary drug-resistant strain of gonorrhea out there. So you definitely want to know, take that bacteria, take that sample from that patient and run what we did in lab and see which antibiotics work against it. So you know exactly which one to prescribe. This is just going to kind of take a look at what we did in lab. So we'll take it a little step further and talk about how to check the results. And we'll do that, of course, on Tuesday. So we're going to measure some in vitro activity. That means outside of the individual, so on a plate, in a test tube, and so on. That's what in vitro means. And again, just like I mentioned on Tuesday, this is standardized. This is the exact way this test is run in labs and clinics throughout the country. Okay, you have to use a specific agar. Does anyone remember the name of that agar?
MH.
It was MH.
Mueller-Hinton.
Mueller-Hinton to agar. So Mueller-Hinton agar is what you need to have, that particular media, because the drugs will diffuse at a certain rate throughout that media. Okay, so using that media, the rate of diffusion is set, and then these standardized tables can be created from that. So here we can see results. So again, everybody created a lawn. So you covered the entire plate with bacteria, right? Maybe Amanda had E. coli, maybe Barbara had staph, and maybe Jimmy had Pseudomonas. So everyone had a different bacteria, and then you stamped it with those 6 drugs. Okay, we used different ones that are up here, but chloramphenicol was one that we did use. So we stamped it, put it in the incubator, and yesterday when I came in, results, they're already done. Okay, they're already ready to read. It usually takes like 18 hours, okay, until you get results. And looking at it, what you want to do is you want to measure the zone of inhibition. The zone of inhibition is the clearing around the disc. You're going to see that it's going to be circular, and you would measure from one edge straight across to the other edge. So you measure the diameter across the circle. Up here, you can see what results would look like. So here you can see everywhere the bacteria grew, and then you just measure from edge of disc growth, or edge of bacterial growth through the disc to the other edge. These two drugs, if you were to trace this out, this one would end right about where the arrow is here. This one, if you were to follow it, would end here. There's this extra space of clearing in between the two. That just means those two drugs work better together than either one does by itself. Okay, there's a name for that. It's called the synergistic effect. Oftentimes, if a drug has, intermediate effect, kind of like so-so effects on a particular bacteria, oftentimes they add a second drug. Okay, that'll help it work better. So that's called synergism or the synergistic effect. So then what you're going to do is we'll just use chloramphenicol as an example. So let's say you measure straight across and you end up with 17 millimeters. Okay, so you end up with a value of 17. So now you're thinking, okay, let's say Anna did it, and Anna's like, I got 17. What does that mean? What does 17 millimeters mean? Well, 17 millimeters, you would need to look at a table. The table says R is anything less than 21, S is anything greater than 21. Anna ended up with 17, so she would say that that bacteria is resistant to chloramphenicol, and she would assign an R for resistant. So that means this would not be a good choice. Even though it looks like there's a nice clearing, okay, according to these standards, it's not good enough. Okay, it's not good enough of a killing. Over here, we can see that these results, okay, this would be sensitive. So here maybe, Barbara did this and ended up with 27. Okay, 27 would be sensitive, susceptible. Okay, so it's able to easily kill it. You can't just look at the size of these because here's intermediate resistance, but it's smaller than the resistant one. Like, you wouldn't think that, but again, every single drug is different. Every single drug has its own set of standards. That's what you have to keep in mind. The table looks like this. Okay, so here you see the values for susceptibility or sensitive. Here you see the values for resistant. Anything in between the two is intermediate. In your lab book, they actually give a third column for intermediates. Okay, just so there's no, mistakes. Okay, so it makes it pretty clear as to which category it is, susceptible, resistant, or the intermediate. So sample results, bacitracin right here. You end up with 15 millimeters as an example. What would you assign it? Okay, so here you're looking at 15 is greater than 13, so it would be sensitive or susceptible, and so on. So that's what you guys will do on Tuesday. You'll measure all 6 of your zones, look at the table in your lab book, and then assign an R, an I, or an S. Okay, and then you can say, for that strain we have in the lab, of those 6 drugs, which are the best drugs to kill it. What we're going to see is one of the bacteria that we used. It's really, really hard to destroy. I'm trying to think. I think basically every single drug is resistant, maybe except for one that is intermediate. Does anyone have an idea which bacteria that would be from Tuesday? Pseudomonas. Yeah, that Pseudomonas is really hard to kill. The strain of staph. I mean, yeah, there's, a form of staph known as MRSA. We don't have that in the lab. We're not working with MRSA in the lab. The strain of staph we have in the lab, after doing this particular procedure, those 6 antibiotics can target that staph pretty easily. They work very well against the strain of staph we have in the lab. Doesn't mean they work very well against every strain of staph, just the one that we have. Here again, you see some sample results. If this lawn is from bacteria A, this lawn is from bacteria B, which bacteria is the easier one to kill? A, because you can see more clearings. Of course, you'd have to do, the measurement and look it up in the table to see, the results for these, but you can look over here and be like, all right, there's a couple of drugs that do nothing. This one really is nothing, very slight, but that's going to end up being resistant. So you can see there's a couple of good choices. Some of the drugs will stain the agar a little bit, just like some of the compounds you used with the antiseptic disinfectant lab, it'll stain the agar a little bit too, but you still see that nice clearing. Another thing you can do is you can do an MIC test. MIC stands for minimum inhibitory concentration. Okay, MIC, minimum inhibitory concentration. That means the minimum concentration, the smallest concentration used to kill. Okay, what is the smallest concentration of this drug needed to get the job done? Because if you were prescribing this drug, you would not want to prescribe a super high dosage if you didn't need to. The higher and higher you go with the dosage, the more and more likely you are to cause some side effects, toxic effects to the host. You want to minimize those effects with the host, so you want to prescribe the smallest amount needed to get the job done. So that's your MIC. Minimum inhibitory concentration. You're welcome. So in order to figure out this MIC, here's what you would do in the lab. Okay, so you would have a series of test tubes. Okay, so within these tubes, you would have the same amount of bacteria in each tube, and then you would add different amounts of drug. In this one, you add no drug. So when bacteria grows, it grows nice and cloudy, like you see right here. So this yellow would indicate cloudiness. This one, you added 0.2 micrograms of the drug. Then you added 0.4, you added 0.8. So you're adding more and more of the drug. Here, at 6.4 micrograms, you see that there is no growth of the bacteria. So according to this series, you would say 6.4 micrograms is the MIC, the smallest amount needed to kill. Why would you give a patient 12.8 micrograms when 6.4 is going to get the job done? The higher you go, the more likely you are to have some toxic effects with the host. Now, there's a big gap in between these two. So of course, if you were in the lab, you'd be like, all right, I'm gonna run, 3.8, a 4.2, and try and pinpoint it a little bit more. Just based on these numbers, say, all right, that's, the minimum inhibitory concentration. What about this series of tubes? What would it be here? 4. It's just the first tube, which you don't see bacterial growth. Okay, the lowest concentration of the drug that gets the job done. Right here is 4. What about this one? 6.4. First tube that's clear, right here, 6.4. Minimum inhibitory concentration, 6.4. Yes?
Could it look pretty clear and still have some bacteria in there, though?
If it still had some bacteria in there, it wouldn't be the MIC. You would want to get a clear one where you don't see any bacteria.
Yeah.
Okay, so you vortex it really well, just to make sure there's nothing in there.
I'm just saying we can't see bacteria with the naked eye, so I'm wondering if there's still possible there could be bacteria, even though it looks clear.
But the thing is, you would have it growing in the incubator where it would multiply, and if you have it in there for long enough and there's nothing visible, it's not multiplying, nothing in there. Okay, one more measurement we'll talk about, one more thing that can be done is called the TI. Okay, this is a therapeutic index. I'll put an example on the board just so you guys can see it. Wrong way with the screen. Okay, so looking at the TI, we are going to be looking at a ratio. So looking at a ratio of the dose of the drug that would hurt us, okay, toxic to us. So up here, I'll put TI is ratio of the dose toxic to us on the top, okay, compared to its MIC, its minimum effective dose, okay, compared to the MIC. And I'll say kills bacteria. So let's do this with 2 drugs. So here's drug A. So for drug A, let's say we have 50 micrograms toxic to us. That means 50 micrograms is going to cause some harm to the host. And the MIC, what you need to kill the bacteria, is 5 micrograms is the MIC. What is the TI? What is the therapeutic index of drug A?
10.
10. So we'll say TI equals 10 for drug A. Great. Now let's look at drug B. For drug B, let's keep, when we look at the TI, let's keep the 50 the same. Okay, 50 micrograms is toxic to us. Okay, so 50 micrograms would hurt us. But now let's say instead of just 5 micrograms killing the bacteria, maybe you need 25 micrograms to kill the bacteria. What would the TI be for this? 2. Okay, so our TI here is 2 for drug B. The smaller the ratio, the greater the potential for some type of a toxic, reaction. So that means the smaller the ratio, like this one, is bad. Bottom line, you want to have a high TI. It rhymes. High TI is what you want. So this drug A is the drug of choice. It has a higher TI. You want a high TI. I mean, you can look at these numbers and figure it out without even doing the math. Because, 50 is going to hurt us. This one you have to take 5. You're much further away from 50. This one you have to take 25. You're much closer to 50. Yeah, this one's going to hurt you more. So you can think about it that way, but you do the math, look at the results. The high TI is what you want. The lower the TI, the more, toxicity that may be involved. What if it doesn't work? What if this drug treatment is ineffective? A few reasons that it can happen. Maybe that drug didn't get to where it needs to go in the body. Okay, maybe it, isn't meant to target that particular area. Here's the big one. Maybe there's a few cells of that organism that are actually resistant to that drug. Didn't appear when you did, that Kirby-Bauer, but there's some that are actually resistant. Those resistant ones are not going to make their presence until you begin antimicrobial treatment. The antimicrobial treatment kills all the ones that are easy to kill. Those resistant ones will start growing once everyone else is out of the way. Then you would need another drug to target those. Okay, so that can happen. Another one, is caused by more than one pathogen. So a different strain is in there as well. Not just a few resistant a few resistant cells to that same culture. With that same culture, here you have a different organism as well, which wasn't apparent at the time of testing. Let's do a concept check. So, of course, you should be able to list and describe a handful of characteristics of what would be the perfect drug. Again, we saw it was affordable, it's going to target what it needs to target, it can diffuse nicely within the body, and so on. All right, true or false? And this first bullet point, that goes back to that first table I showed you where I had about, there was like 8 to 10 characteristics on there, so just know a handful of them. All right, true or false? Cephalosporins have all the characteristics of an ideal antimicrobial drug.
False.
That's false. Okay, again, there's no drug that has all those characteristics. All right, what is the name of the test that we did on Tuesday where we stamped it with those discs?
Kirby-Bauer.
Kirby-Bauer. Okay, so that is the Kirby-Bauer test. True or false? An antibiotic with a TI of 4 is less toxic than one with a TI of 40.
False.
False. It's the exact opposite. The higher TI is less toxic. So the one with 40 is less toxic. Okay, it's a better choice. All right, true or false? Once antimicrobial susceptibility. So once you run the Kirby-Bauer test, you know exactly which drug would target that bacteria. Choosing the right drug is an easy process. What do you think about that? I see some head shaking yes, I see some head shaking no. Basically, it's never really an easy process because, yeah, you might know which drug might target that organism, but what about the patient? What can they handle? What's their age? What's their immune status? You know, what's their kidney function and so on. There's a lot of other questions involved with it. So that would be false. Nothing about these antibiotics are going to be easy. All right, let's just cover a little bit more, and then I'll give you guys a break. So let's talk about what they target. Okay, so what these drugs can target. So, you know, earlier in the semester, we learned all about bacteria. We learned all about what bacteria have, what they do. So when you design these drugs, you know, when there's a scientist in a lab designing a drug, they're targeting what these bacteria have or what these bacteria do. Now, as far as a virus, it's very difficult to come up with an antiviral because they don't have much and they don't do much on their own. A lot of antivirals are going to hurt the patient because they target what the virus does, and the virus does its thing within patient cells. Okay, so they're very harmful. A lot of drugs out there are going to target enzymes. Remember we learned about competitive inhibition? Okay, where that drug can mimic a substrate, bind on to the active site, shut down the enzyme, which kills the bacteria. That's how a lot of them work. There's scientists in the lab right now saying, all right, this bacteria, what enzymes does it use? You know, what can we develop as a drug to target those enzymes? Bottom line, they need to be selectively toxic. Okay, so they need to hurt the bacteria, but not hurt the host. Here are the 5 common structures or functions that most antibiotics are going to target. We're going to go through and I'm going to talk to you about a few specific antibiotics. I'm very specific with what I want you guys to know for the next test. It's just up to you to sit down and study and make certain you learn, you know, these specific drugs. It is not in our course competencies to teach you every single antibiotic out there. Those of you going into nursing will have a course called pharmacology and you'll get all of that in there. In here though, I feel it's important to at least make you all familiar with a number of these different drugs and how they work. Okay, so that is what we are going to do. A number of the more common ones, you at least know their mode of action. So let's start looking at this. Here are the 5 structures or functions that these drugs target, and we'll talk about a few drugs in each category, each of these 5 categories. This picture right here shows, like these drugs right here, they all work against protein synthesis. They're all going to target the ribosome, maybe the large subunit, maybe the small subunit. Okay, so you can see the names of the drugs. These are going to target the synthesis of folic acid. Okay, over here, these are going to target the cell membrane. These are going to target the cell wall, and so on. This little table, here you can see the spectrum range of drugs. So looking at like the sulfa drugs, for example, they're a relatively narrow spectrum drug. They just target, as you can see right here, gram-positive bacteria and a few gram-negative bacteria. Tetracycline's much more of a broad spectrum antibiotic. They're going to cover a much wider range. The polymyxins, they work very well against gram-negative because polymyxins target cell membrane. Gram-negative have that outer membrane. Polymyxins target that outer membrane. So they work very, very well against gram-negative. Okay, so here we're going to start. We're gonna look at all 5 of those processes, okay, all 5 of those antibiotic targets, okay, and with each target that we talk about, I'm going to show you a table. You can see them down here, I'll blow them up a little bit bigger. These tables are right in your textbook, and I'm actually circling the drugs I want you to know. Yes, you need to know the ones that I circle and that I mentioned in class. Okay, so we'll get there in just a moment. First off though, let's take a look at these drugs that work against the cell wall. Cell wall is made up of a molecule called peptidoglycan, right? Okay, so it's made up of this PG layer. And as bacteria exist, you know, they want to maintain a really nice, healthy PG layer. So if it starts to break down, they fix it. A little more breaks down, they fix it. The younger the bacterial cell is, the easier time they have fixing it. The bacterial cell gets really old, just like we talked about with gram staining. Cell wall starts to break down, they have a hard time fixing it, eventually the bacteria die. So drugs that target the cell wall, drugs that target the bacteria's ability to fix the cell wall, they work so much better on younger bacteria than older bacteria, because older bacteria aren't actively fixing their cell wall. So if the drug targets the bacteria's ability to fix the cell wall and the bacteria isn't fixing the cell wall, the drug's not going to work. Okay? If the bacteria is actively fixing the cell wall and that's what the drug targets, the drug is going to work. Okay, so keep that in mind. So it works very well on the younger, the newer bacterial cells, because they are the ones making more peptidoglycan, fixing their cell wall. Here we see a little schematic of this. So right here we're looking at a bacteria. Okay, so it's got its PG cell wall. You expose it to a penicillin, let's say. Okay, cephalosporins or penicillins work really well against the cell wall. They're good cell wall inhibitors. And all of a sudden there's little weak points that develop and the bacterial cell can't fix it. They can't fix it because the drug is around. If that drug wasn't around, they would fix it and be just fine. If they can't fix it, eventually, okay, that cell is going to be destroyed. Okay, so it is going to burst. Okay, and the bacteria dies. Okay, thanks to having that antibiotic present. So let's look at this. Okay, so this is table 12.4 in your book. The way that this table is set up, here we can see the drug class, the mechanism of action, subgroups, and some little blurb on uses or characteristics about it. So for drugs that target the cell wall, I want you to know that the penicillins target the cell wall. That is their target. Penicillins target the cell wall. Do you have to memorize all of these subgroups? No, you don't, unless I circle some of them. But it's good just for you to know that all of these cillins are in this group. Okay, ampicillin, amoxicillin, methicillin, azocillin, they're all in this group. Okay, so the penicillins target that cell wall. Cephalosporins also target the cell wall. Cephalosporins primarily are going to be administered via injection. Okay, they're not absorbed very well within the intestinal tract. Okay, so far penicillins and cephalosporins work really well at targeting that cell wall. You'll notice all the cephalosporins, look at the names of them. They all start with the term ceph. Okay, so they're all in that class of cephalosporins. Carbapenems. Carbapenems, there's a number of organisms out there that are finding resistance to carbapenems, and it's getting kind of scary. Okay, there's carbapenem-resistant enterococci, the CREs, okay, Enterobacteriaceae, so it's getting, this one's getting a little bit scary. Okay, so the carbapenems. And then over here I circled a couple miscellaneous drugs, so bacitracin and vancomycin. So there's 5 that I want you to know that target the cell wall. Okay, so bacitracin, neomycin, and polymyxin. Okay, these three drugs, these are the three drugs that you are going to find in Neosporin, or if you were to buy it generic, it would be called triple antibiotic ointment. These are the three. So bacitracin, neomycin, and polymyxin. Okay, and I just mentioned that just because bacitracin is one of them that I have circled up here. You see it's the main ingredient in Neosporin. Next up, we have drugs that target nucleic acid synthesis. What does that mean? What's an example of a nucleic acid? DNA, RNA. So they're going to target the making of DNA, so DNA replication. They're going to target the making of RNA transcription. So any process along, those reactions, they'll target. So here we go. Drugs that target DNA or RNA, the fluoroquinolones. Okay, fluoroquinolone. Nalidixic acid, maybe one you've heard of before. I'm trying to think. Sometimes we use that when we do the Kirby Bauer. I don't think we used it this semester. Levofloxacin you may have heard of before. Cipro's on here. Okay, so they're going to target DNA or RNA. These are all fluoroquinolones. But again, you don't have to memorize the subgroups, just the class, fluoroquinolones. Here we can see, here looking at these guys, they're targeting helicase. We know helicase unzips the DNA double helix. It's a target for some drugs. Next up, so just fluoroquinolones for DNA, RNA. So now looking at protein synthesis, translation. There's so many targets for protein synthesis. It's pretty complicated. There's 2 ribosomal subgroups they can target. They can stop the mRNA from, being read by the ribosome. Lots and lots of different targets. It's a really nice target because what is the size of our ribosomes? Overall size. Does anybody remember?
ADS.
ADS. Yep. So they're ADS in size. What is the size of bacterial ribosomes? 70S. So because they have a different size, they're going to have different regions that these drugs can target. So you can develop a drug that targets bacterial ribosomes, doesn't hurt ours. Okay, so we can see that here. That's awesome. However, we know in our mitochondria, we have bacterial-sized ribosomes. There's 70S ribosomes in our mitochondria. So some of these drugs may target our mitochondria, and then of course energy production, ATP production is a little diminished, but really good target nonetheless. So let's look at drugs that target protein synthesis. We have the aminoglycosides. Okay, aminoglycosides, streptomycin is an example. Relatively broad spectrum. And then the tetracyclines. Tetracyclines, again, relatively broad spectrum too. Okay, macrolides as well. So here we have erythromycin, I think is one we use in the lab. Z-Pak is on here, azithromycin. Targeting the cell membrane. So targeting the cell membrane, they're going to work very well against gram-negative bacteria. We have the polymyxins. Polymyxin B, most prominent one out there. So work very well against gram-negative. They did develop a new one, called daptomycin, which can target gram-positive as well, but you look at the polymyxins primarily targeting gram-negative. That's also in Neosporin. All right, last one we have, last category, we're looking at folic acid synthesis. Folic acid, don't think about it in the world of A&P. You may have learned about folic acid already. Folic acid for these bacteria is necessary to make DNA, to make RNA, to make proteins. Okay, they need to make this folic acid. So if a drug stops them from making folic acid, they can't make those nucleic acids. They can't make enzymes. They can't make proteins. So another awesome target. So scientists figured out the reaction that bacteria use to make folic acid. And they developed these drugs to stop, one of the enzymes or two of the enzymes along this pathway. There's so many different steps along this pathway. So they developed drugs that target these steps. So here, let's talk about how it works. This looks scary, so let's just make it a little less scary. Let's just show you the reaction first. You don't have to memorize it, but what I want you to see is here's our substrate. PABA is our substrate. That's what we start with this reaction. You can see they make a number of intermediates. Then eventually they make the folic acid, and it's used to make, the bases, amino acids, so they can make DNA, RNA, protein. Great. We can stop them from making this. They can't make these, and they're going to die. So scientists develop these drugs. Sulfa drugs will stop this part of the reaction. Trimethoprim would stop this part of the reaction. Commonly, you would be prescribed both of these together because they work better together, because what if some of this intermediate got made? That's okay. You have trimethoprim to stop it here. Okay, so oftentimes they're given together. So now, the way that it works is if you look over here, here's the sulfa drug, here's the substrate. The drug is made to resemble the substrate. It just has a little different change in structure. The part that binds to the active site is the same, however. So if you look at this reaction here, here's your big green enzyme. Here's the active site. Here's your normal substrate. This guy, your PABA. It binds, reaction goes, makes folic acid, makes everything else, happy bacteria, right? Here, show you that portion. Here we have a few sulfa drug inhibitors present, but look at the ratio of drug to substrate. If you have two that have relatively the same shape that fits with the active site, who wins? The one that's present in greater numbers. In this scenario, substrate's going to win. Reaction carries out, bacteria makes folic acid, bacteria is happy. This is not enough of the drug. So you would need a higher dose of the drug, more potent dose of the drugs. Now there's more sulfa drug inhibitors. Now they are going to win. Sulfa drug inhibitors win, reaction stops, bacteria can't make folic acid, bacteria is not happy, bacteria dies. So you have to have enough of it present. So of course the two drugs, you have sulfonamide, so here you're looking at the sulfa drugs, and trimethoprim. Okay? And a little arrow I just put there because this is going to be, given in conjunction with the sulfa drugs. Okay, so they kind of go hand in hand, and together they do a nice job of stopping the production of this folic acid. Okay, sulfa drugs and trimethoprim. Viruses, what about viruses? You can take all the antibiotics under the sun, they're not going to work against viruses. Why do physicians oftentimes prescribe an antibiotic when a patient has a virus? Why do you think that is?
Out of the office.
Because the patients complain a lot? That's one big reason, yeah. You're a physician and you have, a couple dozen people coming into your office having, coughing, and you can tell that there's a cold going on or whatnot. It's a viral infection, and they're all complaining that I'm not leaving until I get some type of an antibiotic. And the physician's like, all right, and just gives it to them. Sometimes they just don't want to deal with it. Why, maybe they don't want to give a microbiology lesson to every single patient that comes in their office, because a lot of patients don't understand that an antibiotic is going to do nothing against a virus. So the patient's going to walk out with this antibiotic, start taking it, And eventually their cold's gonna go away and they're like, see, I'm glad I went to the doctor because I got this antibiotic and it knocked out my cold, you know? And the doctor wasn't right, it worked, but